Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is about transcriptomics to translation, and to help us with this topic, we are joined by Inga van den Bossche. Inga carried out her undergraduate and master's degree in materials science and engineering at Imperial College London and is now a doctoral student since 2022 in Professor Molly Stevens' lab at Oxford, and she's researching cellular interfaces and innovative biomaterials for regenerative medicine. Welcome to the podcast, Inga.
Thank you so much. I'm really excited to be here and to talk more about my work.
Awesome. Yeah, I'm really excited to have you on, and let's get into it. Firstly, I'd like to ask why you got into material science, and then what sparked your interest in spatial transcriptomics.
Ooh, those are both really good questions. I think starting with why I got into materials science, I didn't actually know I wanted to do materials science till I walked into the department at Imperial. And they had an open day, and in this open day, they were actually looking at a butterfly wing under an SEM, so a scanning electron microscope. And so this shoots electrons at material, and because of the contrast, you can really see very clearly at extremely high resolution, so at the nanoscale. And they allowed me to kind of play with this microscope and put it into focus, and all of a sudden I saw these beautiful ridges. And they told me, this is what makes the butterfly wing blue. And I said, wait, it's not actually blue? They were like, no. And I was like, this is like art, but it's for scientists. And I think I was bought, and I was like, yeah, I think I'm going to do materials because it's art, but for scientists. There's a lot of, really beautiful aspects to it.
Wow, that's so beautiful. The way you described that. I didn't know that was why they were blue.
Not all butterflies,
Yeah.
but really big butterflies from the Amazon forest, yes. Mm-hmm. So it's because of the structural configuration at the nanoscale. So these ridges make it blue. Mm. And I think, so I guess imaging kind of caught my interest as an undergraduate. And then I went into materials. I did look a lot at biomaterials as well. I looked a lot at things at the nanoscale, so nanomaterials. And then I ended up in a PhD, and in the PhD, the first thing that caught my eye, I would say, is really this idea of looking at cells under a microscope and then also being able to distinguish between cell populations. So if you look at transcriptomics, and especially spatial transcriptomics, the exciting aspect of it is being able to look at the profile of a cell, being able to look at the entire transcriptome, so all of the RNA that's being expressed at that point in time, and image it. And so based on all of the different transcripts that a single cell expresses, it gets a unique identity. I wouldn't call it a fingerprint because it's a snapshot in time, but you could think of it as a profile of a cell. So if I were to take a picture of you right now, that would not be the same as you tomorrow. But in the context of you being a person, you're still going to say kind of the same. And I think it's similar with cells in the spatial context. So when you're looking at, say, for example, an organoid, you can differentiate between cell populations. And you could say, oh, this is a unique cell type based on everything that it's expressing at that point in time.
That's such a cool technique. And how does your research and I guess the Stevens lab then use this cell profiling and the kind of research they do? I understand that they create these 3D, some projects involve creating 3D tissue engineering scaffolds. Could you tell us a bit about that, or if they are used in that context?
Yeah, absolutely. So I think that's a really interesting part of the group. So our group is divided into 3 different groups. So we have subgroups. So we have therapeutics, diagnostics, and tissue engineering applications, or soft materials. And I would say that our group is super interdisciplinary, so it can fall into different categories. But for example, if you want to look at tissue engineering applications, one aspect of like what we think about is cells are cultured quite often in a flat petri dish. But actually, if you culture a cell on a different material, it's going to have a different interaction with that material depending on the strength of the material. And so you can start to think about differentiating cells by growing them on different materials. So if you grow, for example, a stem cell in a really soft environment, it's going to feel like, ooh, like I'm in a brain environment. So then it's going to try and mimic that environment or feel more aligned to follow differentiation into a neuron. Whereas if you put that same cell on a really hard substrate, it might be more inclined to differentiate into a bone cell. So that's the type of interactions one can start to look at when thinking about, culturing cells and materials in these interfaces. And then where you can start to use transcriptomic experiments in this is to understand, well, why is a cell expressing becoming a bone cell in this context? Or is it truly becoming a bone cell? Is it just that it's expressing bone-like features, but is it actually a bone cell? And being able to then sequence a cell in that context is extremely powerful, because you can start to say, well, actually, the way that we are culturing these cells, yes, we are arriving at this beautiful final bone material, or no, we're not there yet, and to get those feedback loops. That being said, it's a very expensive technique, so it's not something that can be done every single day, but I think there's a lot of exciting opportunities.
Yeah. And so do these materials kind of define or guide the differentiation of the cell, or is it more about, like, do they activate the genes that make it go into a bone or a neuron, or is it some other factor, like what you feed, the medium that they're in, that cause them to differentiate in a specific way?
Very interesting question. I think actually it's both. They can be complementary to one another. Right now, I think in a lot of cell cultures, a lot of growth factors are introduced at certain time points to really facilitate that differentiation. And I also think that the structural element can influence the functional expression of what's happening inside of the cell. So there's definitely synergy there. I mean, when you think about inside of your body, if you put stem cells in a cartilage implant to try and regenerate, those cells are going to feel their local molecular environment, like what they're interacting with, but they're also going to feel their environment in terms of like how they're actually located. So if they're located on a scaffold, or how they're interacting with their neighboring cells and with their extracellular matrix as well.
That's really cool. Yeah, it's really interesting to highlight that the physical component of cellular identity, because I think a lot of scientists focus a lot on the molecular identity and how that is guided molecularly, but not so much, especially in terms of neuroscience, from what I know. There's really research lacking in terms of the physical processes that guide that defining cellular identity. So really interesting that you mentioned that and how much it shapes what type of cell they become. I find transcriptomics super cool and interesting, this idea of defining cell types based on the transcriptome. So this is quite a general question, and I know no one has the answer to it, but in your experience working with spatial transcriptomics and transcriptomics in general, how discrete do you think cell types really are? Because surely there is this level of continuity when you look at the transcriptome.
Absolutely. So maybe just to clarify, I don't actively work with transcriptomics, but it's something I've extensively looked into and I'm super excited about. I work a lot with what one consider more the probes of transcriptomics. So I've actually gone down all the way into the DNA, RNA level of things. I'm really starting to think about how can we design new probes that are more accurate and more specifically targeted, as opposed to taking the whole transcriptome.
Well, it's just honestly coming from personal experience, because I remember personally in my undergrad we were learning about cell types, and in the brain you have excitatory neurons, inhibitory neurons. Those inhibitory neurons can be split into different types and based on various things, but now there's really this push for transcriptomics to really define these cell types and use the transcriptomic data to understand if this is really a true cell type. But then I remember one of my professors were like, But the cells don't know what they are anyways. Like, a cellular identity is just something we use to define them in order to study them better. And we were all kind of blown away because we were like, oh yeah, this isn't set in stone. This is all subject to change, and it's all humans defining them. And then when you look at a lot of the RNA sequencing data and everything, like, the methods that are used are all cluster-based. And so the analysis, as far as I know, does affect, how many types you can really define, because there's so many different RNAs that can be expressed in these cells. And so where do you kind of draw the line, and how discrete is that cell type classification?
No, I think it's a really good point. And there's also the added factor of time, is that cells change over time. So yes, I agree actually with your professor when you said that we're putting a profile on a certain cell, and it's about like how coarse grain or how fine grain are you going to look at the subtleties between different cell types and say this is a unique cell type, or actually is it really a unique cell type, or is it just some noise, or is it actually that this cell type is in this particular phase at this point in time. And so maybe to answer your question, or not to answer it, because it's a very difficult question to say, like, what are the boundaries between one specific cell type and another specific cell type, and how specific do you need to get? I would say you have to get very specific, and I would start to look at different, like, there's generalizable populations. So you could say this is a general profile of this type of cell we would expect, or this is a general expression profile of what we're expecting this cell in this context to have. But then taking into account the time factor, and then potentially also looking at different omic techniques. So potentially correlating to like proteomics, for example, or correlating to methylation of DNA, or looking at like the chromatin orientation itself. So really starting to integrate different omic techniques, I think, can give more, and this is very difficult, but can give more confidence in, like, actually we see that if this RNA is expressed highly upregulated quite often, it might correlate to a protein, but the protein is very lowly expressed. So sometimes these data sets correlate, sometimes they don't correlate, which adds, again, another layer of complexity. But coming back to the cell type question, I've also thought a lot about gradients and how this changes over time, and when you define, for example, especially cartilage is a very interesting material, because it's a gradient both from the nanoscale to the macro scale, but then it's also a gradient in terms of these cells are in this environment, and from one side there's a soft tissue and another side there's a hard tissue, but the interface of cartilage it's hierarchically integrated. It's really nicely done, and it's a material that is still very difficult to mimic from a material side of things. And so one of the questions that I've had a lot is, how can we start to understand what these cells feel in that soft environment versus that hard environment, and what about along the entire gradient, like how are they changing?
That's so interesting. And also, could you explain a little bit about what the difference between spatial transcriptomics is versus just any transcriptomics? What's the spatial aspect of it?
Yes, so spatial means looking at the tissue without dissociation. So all single cell transcriptomic techniques, or NGS, what single cell does is it takes a tissue, it dissociates it, so it separates the cells from one another, and then it sends these cells through microfluidic tubes where they're encapsulated with emulsion, and they're also barcoded when this happens. And so then you can start to say, okay, we now know this cell is, we now can check if this cell is one particular cell, and then it can be sequenced. But the disadvantage of this, and really what is the unique advantage of spatial transcriptomics, is that you don't have to dissociate your tissue. So for example, when you look at cells, they have things that are being expressed within themselves, but they also have cell-cell interactions that are extremely important. And so being able to, or when you look at, for example, axons in the brain, these are very elongated cells. So being able to see where all of these cells are interacting with one another is very difficult in a single cell experiment, because you might not be capturing the full cell, or parts of those axons are being cut off. So in a spatial context, you can really start to minimally perturb the tissue and look at it in its context. So I think that's the really big difference. That being said, single cell is much higher resolution, but there's exciting breakthroughs happening on both ends, and I think they're both super cutting edge fields.
Yeah, bringing in the topic of interdisciplinary, this emphasizes a bit of a parallel between spatial transcriptomics and I guess one of the focuses of electrophysiology and neuroscience, in the sense that it's about the cell-to-cell interaction, and not only the cell-to-cell but the whole circuitry and how different cells are wired up in the brain, but also it's not only just about the molecular composition of these cells but their function, and so it's just really interesting how both with neuroscience and it seems with molecular science, there's this push for understanding the greater dynamics of the system, and that there's a lot of value in understanding, fine single cell and single gene resolution, but how these are all connected with each other is also really important. So it's interesting to see this happening in other fields as well.
Yeah, absolutely. I think also you mentioned this idea of long range order interactions. It's a really interesting question. And like going back to this idea of making environments that allow cells to feel very happy in them. So thinking about synthetic materials and organ chips or like organoids, even how they're being grown and how you're going to perturb them or provide that right environment. There's a lot of interesting studies that are being done at the moment into looking at correlating different organoid chips. So emulate, for example, is a really good example of this. But also a lot of research is happening on this front, is being able to see, well, actually, how does the gut-brain interaction work? Because that's also a very interesting and a very strong connections happening there as well, and they're not right next to each other. So that's not something that you could look at when you're taking a biopsy from a patient, for example, directly under one microscope. But being able to look at both those samples and being able to correlate across them is also very interesting. And the same can be done with organ chips. Like you could hook up one organ chip from a brain to a gut to study that interaction.
Oh, that's really cool.
Yeah, I haven't actually heard of research directly using organ chips. Could you explain a little bit how these are used, or how these organ chips are generated and used in research?
Yeah, so an organ chip would be something where you're trying to make an environment that mimics an organ inside of the body. So for example, when you're making a lung organ chip, you would try to mimic the lung lining, or try to culture the cells of the lung and try to simulate how the lung stretches, for example, or how it's breathing, or what are the fluids that are interacting with it, or how does the lung lining look. And so it's still, again, a very early stage of research, but I think there's a lot of opportunity there if you can culture your cells really well and really mimic what is happening inside of the body, because then you can start to use this model similar to how you would grow cells in a petri dish, but also considering pressure, forces, fluid that is flowing, so a dynamic environment, which is a big advantage of organ chips, which are these small chips which have microfluidic channels in them as well. And so you can start to use this model and mimic diseased states as well. So being able to look at, well, if I add this into this environment, how is it going to change the cells? Very similar to how you would do with a normal cells in a petri dish, but also considering those forces and interactions.
That's really cool. I never really understood how an organ chip works, and now I have a much better understanding of it. Going back to Clara's question about how discrete are the characterization of spatial transcriptomic and the genes. This reminds me a little bit of an episode that we did with Sydney, where he was looking at converting glial cells into neurons, and that to what extent were they confident that they were being able to achieve that, looking at the molecular and the genetic composition of the cells. So that was really interesting. Going back to your PhD, could you tell us a little bit about the project that you're currently working on?
Yeah, absolutely. So I was initially looking at spatial transcriptomics because I think it's a fascinating field, and you get to color your cells kind of like when you're going in the tube and everything is also colored, so it's very visual science. I really love it. It's really beautiful as well.
Can definitely relate to that. I love my imaging days the most.
So I think it's a really exciting, really beautiful. And so at the moment, I'm actually working a little bit more on projects that are inside of our lab. So we do, as I mentioned, therapeutics and diagnostics as well. So I've realized I really love RNA. I think it's really cool. There's a lot of opportunity in both the therapeutics and the diagnostic space. So right now I'm working on diagnostics using microRNAs, so detecting microRNAs using strand displacement systems, and really thinking about more the translational side of diagnostics, as in like, what do you need to make a good diagnostic? It needs to be cost effective, it needs to be easily accessible, and then thinking a lot about, well, what can we do with diagnostics taken to the next step? So how could you integrate, for example, a diagnostic in somebody's home? Similar to how with COVID we had lateral flow strips, and now there's a lot more opportunity to bring lateral flow strips and this idea of regular testing into the home. So that's something I'm thinking about a lot. And then I'm also thinking a lot about personalization, because similar to, and this actually, the field of spatial transcriptomics and diagnostics, spatial transcriptomics is fundamentally a diagnostics technique, but it's expensive, and you can't take a biopsy at home and look, do I have breast cancer or not? So whilst it's a really beautiful technique, it's still very costly, and there's exciting things happening, like thermal plex is a really cool paper as well. But I think that accessibility is what really attracted me to the diagnostic side of things, and thinking, okay, well, you can look at the disease in a biopsy at a late stage, but what if you could detect a disease at an earlier stage? And what if you could detect it at an even earlier stage, where it's before metastasis, or before, like, you can really start to prevent the disease happening in the first place, or even just checking what is your personalized healthy baseline. So I think a lot about personalization as well, and being able to look at baselines of individuals, because fundamentally everybody is different and has a different baseline of like expression.
Yeah, that's really cool. And I think it's such an attractive thing about science nowadays, and especially personalized medicine is becoming bigger and bigger, and everyone knows that prevention is 10 times better than finding the cure. So if you can catch it early and notice it, then that's really vital. You mentioned microRNAs and strand displacement. Can you tell us a little bit about that? And
yes, absolutely. So when we look at DNA is what your genome is made of. So it encodes for the color of your eyes, how you look like. That's the DNA defines how you are. Then to make this DNA become you, it needs to be expressed in some form. So the form it's expressed in is RNA. So one could think of DNA as a double strand, RNA is a single strand of almost the same letters, with the difference being T and U. And so when this RNA is then read, it gets translated into a protein, and then that protein could become hair, skin, it could be an antibody, it could be an enzyme, it could have multiple different functions. And so in this way, from DNA to RNA and protein, there is also reversible ways. We can also go from RNA to DNA back again and have multiple more complex interactions. But RNA is thus what encodes for proteins. And so if you can start to look at RNA, you can start to see, well, if there's a protein that is causing a disturbance in the system, maybe we should tackle the RNA before tackling that protein, or trying to sequester that protein or downregulate it. And so microRNA is kind of like an mRNA in the vaccine, the COVID vaccine, the material the vaccine was made of. It's that RNA, but taken into small snippets of about 18 to 21 or 22 nucleotides. So the same would be letters, 22 letters. And so what I'm trying to do, bringing it back to the diagnostic side of things, is trying to look at, imagine if you had a whole book that you would take and try to flow up a lateral flow strip. So you had a piece of paper and you tried to flow up the whole book. You would want to detect only 21 words or 21 letters out of this book, and everything else gets to flow up the strip. So that's kind of the challenge we're working with, is how can we detect these 21 letters and not detect 20 letters and then have like one letter be off, because you really want to detect that unique sequence.
And so how is that going? How have you been going about carrying out that research?
Going back to, you mentioned this strand displacement systems. So the idea is that we have a probe which is semi-stable, but it has a little arm, and that arm is not attached to anything. And because this arm is free, when the final target comes in, it gets to displace one of the strands that is already present. So imagine if you have a short DNA, but a small end of this DNA is free. And so when the final target comes in, it actually is very energetically favorable to displace one of the strands, because the final target is the same length as the backbone strand. So it's always going to be energetically favorable, always very high probability, unless you're starting to play with concentrations and different things. But in a lot of cases, as long as you have this arm, or what we could call a toehold, you can start to displace this final strand, and so hence capture your probe.
That's cool. How easy is this, and how easy would it be to implement it in, like, for example, you mentioned a COVID strip, and just get it out to market and have people be able to use this and maybe have a bit more of like personalized medicine, like you mentioned?
Yeah, I think there is still a lot of barriers to go, especially when you're thinking about translation, because it's not just being able to do something in the lab, it's actually being able to scale it up to the volumes that are needed if you need to treat every single person in the UK. It's also being able to make it in such a way that it's cost effective. So does it make sense to have it per lateral flow strip? And then also, is it stable over time? Because if it's in the lab only stable for 1 week, but you have to ship this to individuals, their homes, that's an added factor. And then also it needs to pass a bunch of regulatory tests. So does it actually perform as well as it does in the lab in a different setting, or when you're scaling it up? Is it GMP manufacturable? So grade manufacturable. And also thinking about how are you going to roll this out? How are you going to make many of them? And then the timelines for diagnostic are still quite long, I would say.
Yeah.
And how easy is it to kind of translate this across different RNAs you want to detect?
Exactly.
So how easy would it be to redesign it for different viruses?
That is a good question, and something I'm also thinking about. I love the idea of modularity. It's something I'm working on as well, because if you can make a system that is modular, it means you can have a plug and play system. So if you imagine you have an input and your system works independent of the input, as in you can change your input and the system will still work as such, that would be really cool. So that's something I'm also thinking about as well, is because right now, and I mean, still a lot of techniques is always looking at one thing, but if you can introduce that modularity, you can start to think about multiplexing, going back to the transcriptomics idea of a cell is not solely defined by one expression of a protein or an RNA or like DNA, even, like you need to look at more than just one, especially for RNA and protein. And so the similar idea would be in diagnostics, is that if you can start to track, instead of one microRNA, maybe 2 or 3, to have a baseline comparison, or to be able to say more specifically, like, oh, we have 2 that are upregulated, 1 that is downregulated, what does that look like, and to give a better picture. So something I'm working on as well.
And what is, I guess, your biggest challenge when looking at trying to make this accurate and making sure you're only getting the 21 nucleotides that you want?
I think there's a lot of factors that come into it. We can think about the structure. So it's not just a linear like line, as you could say, with letters. 21 words on a page is straight, but RNA is not straight. RNA has a lot of secondary structures. So how are you going to get this to unfold? How are you going to choose a target that is still going to be stable over time? Because RNA is not very stable always. So that would be one of the challenges, I say, yeah.
And so this is very much to do with the diagnostics. Can you tell us a little bit about the treatment side of things in terms of your research or your research interests, and how maybe these techniques can be applied to the treatment side of it?
Yeah, absolutely. So I think right now I'm definitely working more on the diagnostic side of things. That being said, there's a lot of opportunity in the RNA therapeutics space as well. And so thinking a lot about the sequence design. So how are you going to choose your nucleotides so that there's minimal off-target binding effects? Or going back to this idea of detecting the 21, or it might be a much longer RNA, being able to make sure that you're detecting this on target or that you're binding to something on target is very important. That's one part of it. That's really looking at the molecular part of it and the design side of things. The other part is how are you going to deliver this? Because mRNA cannot be delivered free floating. It could be, but it's going to have quite low efficacy. So you'll probably want to encapsulate this in a sort of lipid nanoparticle or another type of polymeric nanoparticle that can really stabilize it and allow it to go through your body for longer.
Yeah.
So that it's also expressed higher.
So how could that be therapeutic?
How could mRNA be therapeutic?
Yeah.
I see. Like if it's expressed as a protein, that protein is going to be upregulating or downregulating something. Or when you have so you're trying to express a certain protein. So rather than trying to deliver the entire protein into the cell, you would deliver something which is smaller and easier to deliver, easier to transfect into a cell. So that makes it easier because a protein could be quite big, for example.
Okay.
What's more or less like your accuracy percentage or yield when carrying out your experiments?
Very variable at the moment.
Yeah.
I think that's with a lot of research.
Yeah,
is things are always variable, and then you're changing a lot of variables as well to get it higher, or to try something new to modify the system.
Yeah.
So I would say variable.
Of course. I mean, that just to gauge a little bit how we are in the field, and I guess like how soon we could have this be spun out or anything like that. You've mentioned that interdisciplinary approaches are really important to you. What kind of interdisciplinary techniques do you use in the lab, or not even in the lab? I realized that Stephen's group is highly interdisciplinary. So do you apply techniques that you learn from other people?
For sure, 100%. I think our group is really, really cool because we have about 87 people from many different backgrounds. So ranging from
it's the largest
quite big. I think it's one of the biggest groups in Europe.
Yeah.
I'm not going to say the biggest. I'm not super sure, but definitely one of the biggest ones. So I think it's really, really cool because I get to work together. So I'm coming from a materials background, and I get to work with people who are like biochemists or looking more on the computer side of things, so like coding much more. So previously I was working a lot, and still now actually, with somebody who studied computer science in his PhD, and he's helping a lot with simulations of RNA.
Cool.
And looking at the different software. Then it's really great because we have a really big group meeting. So when we have this group meeting, we have surgeons in the group, and they're like, well, actually, does this material make sense in a surgical context? And then we have pure chemists, which are just really amazing and super brilliant, and they're like, oh, we should do this type of click chemistry or conjugate this, and I'm like, whoa. So I think inside of the group, it's a very important aspect. I would also say outside of the group, I'm also collaborating with another professor because you can know a lot of things, and it really depends on the strengths that you bring. So I think being able to work together with people who are really expert in their field is really, really cool. And then for those experts, it's also really cool to be able to integrate and bring new ideas into the field as well. And I think that's one of the really exciting things about interdisciplinarity is you get introduced into a new field, the other expert also gets introduced into a new field, and then the questions that arise are completely different and unexpected questions that maybe you would have never asked in the first place because you're like, oh, but this rule and this is so obvious. And then actually by having that rule be questioned or coming at it from a different angle, you're like, oh, maybe I should look at it this way.
Especially with translation, as you mentioned, like thinking about actual clinicians in the room being like, oh, well, this is what we can or cannot use in the clinic. And that's so important to actually have that perspective, because you could design the most brilliant technology, but if it's not actually useful, what's the point?
Yeah, exactly.
Yeah, absolutely. And I think that we definitely think a lot about translation. It's very, very important, I think. There's two sides of science. One is really the fundamental science, which is extremely important and gives really novel insights, and then the other side is translating that really exciting and cutting edge science. And so thinking a lot about how can you design really cool systems and make sure they are translatable is very important. And I think the more that you think about translation from the start, the more you can build this into a project, or the more you can define these criteria from the start and say, well, actually my diagnostic needs to be stable over the course of 30 days in a 40 degrees Celsius environment. Or this therapeutic, we're trying to make it stable at room temperature, so we don't have to send it in cryo transportation. So being able to think about that outcome from a very early stage, I think is really cool as well.
I think that sometimes we scientists develop things and produce things and then don't really think about the long-term consequences of it. Yesterday we did a cool episode with someone that was focusing on degrading non-degradable materials, silicon carbon bonds, and he made the point of, when designing something, you need to think about the other side of it, and like whether it's biodegradable or not, and in the case of translational research, whether the fundamental research actually can be applied. So this brings me to ask you about nucleates. Is this where your interest in translation stem from, or were inspired by?
And what is nucleate?
Yes, I'll answer both those questions. So maybe for some context, Nucleate is a nonprofit organization run by PhD students and MBA students. It was originally started in Harvard around 5 or 6 years ago. The time flies when you're having fun. And so originally started in Harvard, and then it kind of spun out in the Boston ecosystem. And then during COVID, there was a realization that actually this is such an exciting program that we should just share it with more people. And I think that that was really the first step into bringing Nucleate into the UK as well. And so what it is, it's a program for PhD researchers, so early stage researchers, or people who are taking their first steps into the venture world and into starting a company as well, and to introduce them into, giving suggestions, meeting with advisors. And so we have over the course of a year, we have 6 different workshops where they're about meeting advisors, how to do a spin out, what it takes to do your first hire, what it looks like to have good company culture. And so really helping with those fundamentals and giving advice. Another would be a very important, what should your next step be to get to that phase 1 clinical trial? Or what do you need to do at this moment in time to get you through the next 6 months? Or how are you going to take your first steps out of the lab into the real world? And really building those relationships, and then it accumulates all in a demo day where startups get to present what they've been working on, and it's a really exciting event as well. So I think my translation interest absolutely have grown a lot with being with Nucleate. I would also say that I've been part of the deep tech space for quite a while now. I'd say since my second year undergraduate. And the reason being because when I was an undergraduate and I came across looking at, for example, bacterial cellulose that could be grown into fashion, like used for textiles. I was like, wait, this is so cool. Like, you're applying science, but you're applying it into something that I could wear every single day. Or when you're looking, you mentioned this degradable plastics. So like looking at startups that are actually making these plastics, or looking at like PHA or PHB, so polyhydroxyalkanate. But these plastics that can be degraded by bacteria. And I just thought this was so cool that there was science that was also like trying to make an impact into the real world. And so in my undergraduate, I did, one of my most favorite courses was actually optoelectronics, going back to the butterfly wing. This idea that you can change colors at the nanoscale. And so super theoretical, looking a lot at the quantum scale of things, which is amazing. Like, I really love it. And then also realizing like, okay, this is really cool. How are these materials actually getting translated? What does this look like in an LED lamp? What does this look like? Can we design this lamp better? Can we design something more environmentally friendly? So I think that's initially where my interests in translation stemmed from, is seeing what is the most cutting edge in science. And that's still something I'm super passionate about. And why I do a lot also in the translation of science and helping translate science as well.
Yeah, that's so cool. I definitely think it's so important for people at university and at different phases of their degrees to take what they're doing and trying to translate it if possible and spin out if possible, because if not us, then who? Like oftentimes big pharmas and big companies aren't taking the risks that maybe that we can take just because we're new at it and we're really in close contact with the science and the day-to-day. So I think it's so exciting and it's so cool that you're doing this and that this is happening.
Yeah, I would just say also to your point about the risk taking, and I'm gonna like stand up for all those blue skies researchers out there, because I think it's really a balance. It's equally about like doing this fundamental science, playing around, trying new things, and also thinking about translation. So like really keeping an open mind about like creativity and not saying straight away, well, okay, this is how it's done today, so this is how we should do it today. I think I question a lot, should we do this differently? Is this really the best way to do a lateral flow strip? Like, yes, maybe right now everybody does it like this, but is that really the right way? So I think that it goes both ways. It's about translation, absolutely, and also questioning, always questioning things.
Yeah, that's a really good point. And I think that it makes a lot of sense given what you've talked about today is a lot of like frontier kind of research, like new technologies, and so you have that kind of room for creativity, which is great, because then you don't, yes, in some things you work on like the lateral flow test, there is a set way that people always do it, but in some other things there is a lot of room for coming up with new methods and variations of the method and thinking more about how could we really cater this to our use. So it's really nice to hear how there is that room for creativity that you mentioned.
Absolutely. And when you're thinking about really changing the way things are done, I think you have to think creatively. Especially like going back to this idea of personalized medicine. If it's not been done up till this point, then something needs to change to get to that next stage. So yeah.
I'm really excited to see success of your PhD throughout the years. Is this something that you want to then take and spin out? I mean, that seems like the natural path.
At the moment, we are still very much in the blue skies research side of things, I would say. That being said, I realize I really like science. I think it's really exciting. I also really like being part of the translational scene. I think it's also really cool. So let's see.
Exciting. I think that's the perfect place to end today's episode. Thank you for joining us, Inga. It's been a pleasure.
Yeah, it's been really nice having you on, and really nice to meet a material scientist with such an interdisciplinary perspective.
Thank you so much for having me. I think it's been super exciting to be here, and looking forward.
If you enjoyed today's episode, please feel free to share with your friends and family. Our social media handle is @neuroverse_pod, and you can find the resources and blog posts to our episodes on our website neuroversepod.com. We'd like to thank the Biochemical Society for supporting Neuroverse, a podcast about science and philosophy. This episode is part of our series to disseminate research done in biochemistry.